Yeah, welcome, Svetlana.
Thank you.
Yeah, it's my pleasure to introduce our next speaker, Dr. Svetlana Boroskina. She's a research scientist at MIT, USA. Dr. Svetlana is a physicist and engineer who studies light matter interactions on nanoscale and develops new smart materials and devices for solar energy harvesting, personal thermal comfort, night vision, space exploration, and biochemical sensing. She leads the multifunctional metamaterials research group at MIT, where the ongoing projects focus on the development of sustainable fabrics for passive thermoregulation and antimicrobial protection, photonic metamaterials with structural colors similar to the butterfly wings, optomechanical energy harvesters that work day and night, and photonic biosensor platforms for rapid detection of chemicals and viral pathogens. As the lead editor of the Energy and Environmental Optics Express, Svetlana is helping to disseminate scientific knowledge and encourage the international collaborative effort to address challenges in the water energy nexus, photonics, metamaterials, and environmental sensing. And now she'll be delivering her lecture on meeting and preventing new challenges in biochemical sensing. Thank you. And over to Svetlana now.
Thank you very much for the introduction. Let me share my screen. All right. It's a pleasure to talk to you all about our work on meeting and preventing new challenges in biochemical sensing. So what kind of challenges are we looking at? So there are a few shown right here on my opening slide. And these include challenges with greenhouse gases, in this particular picture, methane being released in the environment. COVID is the obvious challenge I guess everybody is facing right now. And microplastic pollution is another challenge. So this is how the methane that is naturally generated in ocean looks like. And there are a lot of reasons we still don't understand why it's happening and at what rate it's happening. So it's quite important to have some reliable and accessible tools so we can measure it in places where it does get released. And those places are often remote, like what's shown in this picture, and not easy to access. And because the ocean is so large, you need a lot of instruments to actually get reliable data from different places. So microplastic pollution is a problem that is now becoming serious worldwide because just of the sheer amount of plastic that is used by humans, and then a lot of it ends up in the environment as shown in this picture. And this contributes to pollution with microplastic particles, which take very long time to degrade, and nobody really knows what are the long-term health effects of this. And then, of course, our terrible friend for the last years, the COVID-19 virus. So that is an obvious threat that also reminds us that there are viruses like this that can appear again and again. And it's good to be prepared so it doesn't take months and years to launch new sensing tools, and we can deploy new tools rapidly and adapt them to new viruses, because unfortunately, new viruses will be coming and we need to be prepared. So we need to develop universal tools to address these challenges. And these tools are biochemical sensors. So despite those sensors looking very differently and having different functions, all of them share a common framework terminology. So you need to distinguish in a sample a target molecule that may be present in very small amount, and it may be very small by itself, usually is. So it needs to be captured first by some kind of sensitive receptor. The signal needs to be converted into some kind of signal that we can measure, whether electrical signal or optical. So in my group, we work on photonic sensor development. So the signal we work with is light. And usually it's a very weak signal, which needs to be amplified. So we need to design an amplifier accordingly. And then the signal needs to be processed to understand whether it's positive or negative result. It sounds pretty straightforward, but in every realization, it's different and it can get quite tricky because again, a lot of substances we're trying to sense, they're either present in small amounts or are distributed, and you need to be there at the source of where it's being released. So for example, how a typical sensor for methane dissolved in ocean water looks like. So it's actually quite a big piece of equipment, pretty expensive too, because it usually involves near to mid infrared lasers, which are expensive devices and not everybody can afford to have them. And they are bulky, so you actually need also to have a ship to operate them, to deploy them, and then collect the information from the sensors. So we set out to design a new device that would be much easier to operate and much cheaper. So ideally, in the end, it would be integrated with a cell phone. And Morgan, shown here, so she is a student who works on this project. She actually was expected to spend her last summer in the Arctic deploying sensors like this, but because of the pandemic, her trip was canceled. So, and instead she spent that summer designing a new type of sensor that we are now making. So what is the difference here? We need to have a sensor that's highly sensitive and can be operated in a simple environment. So ideally, we don't want to use any expensive lasers, tunable or otherwise, and any movable parts, because that makes design complicated. And we want a small footprint. And to achieve that purpose, we tried to see what's out in the market already. So the only sensor that was designed that fits this description of simplicity and accuracy is a surface plasma polariton sensor. So it's a pretty simple configuration where there is a prism on which we have a thin metal layer which supports the surface plasma resonance. And on the other side, there is a sensitive polymer matrix that selectively captures methane. And it changes the refractive index of this polymer very, very slightly, but enough to detect some changes, spectral or angular changes in this resonance that originates because of the excitation of the surface plasma mode on the metal dielectric interface. So it works, but as you can see, this mode is pretty broad because of the losses in metal, and it's hard to control it because we cannot change the losses in metal. And that's why the sensitivity is limited. And also to detect changes, we do need to vary the angle of excitation or to scan the spectrum. So that adds complexity to the sensor design, and it's not something that you can easily integrate with a cell phone, for example. So what we did, we re-engineered the sensor, and instead of using surface plasma resonance, we built a simple photonic crystal defect sensor, which also features a resonant mode at a certain frequency, which can be tunable by design. But this mode, as you can see by the frequency or wavelength bandwidth, it's significantly more high quality. So it's a narrow bandwidth mode that is easier to track. And if it moves just a little bit spectrally because of the changes of the refractive index, this can generate a very large difference at reflected intensity at a single wavelength. So if we just excite the system and monitor it at that single wavelength, we don't need to have all the additional complexity in the sensor. And indeed, our sensor design operated at a single wavelength that you need to engineer by sensor design and composition, provides much larger sensitivity. So you can see with very, very small changes in the refractive index, and this range of changes corresponds to the actual changes of the dissolved methane that is expected in the ocean environment. So despite the changes being so tiny, we can have reflectance difference almost from 0 to 80 degrees, which is very easy to detect with simple sensors. And this is a practical implementation of a sensor. So we would be just looking at the intensity changes under the excitation by a simple off-the-shelf photodiode and LED. And this is easily detectable in a wide range of changes of the refractive index that are typical for methane concentrations. So we're making them now and to be deployed and tested at the Woods Hole Oceanographic Institute in Massachusetts. So it's a very interesting facility, very close to us, and they have a joint program with MIT. So that's where we can have access to all the unique marine biology and marine science and engineering tools to actually test devices in the in situ environment. So that's the story of methane, and looking at the story that unfortunately we had to live through all this year is testing for the viruses. So, and this testing is also needs to be dramatically scaled up. So this data here on this slide actually old from the fall, but it gives you an idea of the scale, right? How many people need to be tested daily for a reliable prediction of the virus spread. And these numbers are just astounding. So it means we need to be able to make new sensors, which are scalable, affordable, and they can be quickly redeployed to address a new threat if the virus mutates, which is already happening, we see, right? Or if a new one appears. So what's the problem with detecting this virus? It's also like all the biological and chemical objects, it's very small. So this is just the whole capsid is the size of about 100 nanometers, but what is actually inside is even smaller. And the other problem is it's actually very, very similar. It's a look-alike to all the other viruses of its family or other families. So all of them are small objects with very similar refractive indices to each other. So to the when we probe them with light, they actually present the same target, and that target is so small that it's hard to detect it and even harder to distinguish in between different types. And if we look inside of this virus, where the actual distinguishing part of it is stored, so which is RNA, which tells apart now those different viruses from each other, it's even smaller. So if the virus as a whole is about 100 nanometers, then the DNA and proteins inside are even smaller. And if we're trying to probe this virus, so the wavelength of light is a couple of orders of magnitude larger than the size of the object we're trying to probe. And that, of course, is a problem. And the result may be a false negative because for different reasons. Maybe we cannot just capture that one or few target molecules that are present in the sample, or maybe our signal amplification is not enough. So we did capture, but we cannot really see it. So another problem can be false positive, when the sensor does capture something and it generates a signal, but that something is not a target. It's just something random present in the sample, which often happens with human or any other biological samples, right? Whether it's saliva or blood or any other sample. And that is a problem. Another problem with commonly deployed PCR type of tests, they actually can generate false positive because of the how they work, because they are designed to overcome this problem of very small indetectable amount of biological material by amplifying the amount of this material. So at every step of running the PCR, which usually takes dozens of steps, the amount of biological material is doubled. So every time a DNA is replicated, it increases in size, it doubles in amount. And this is, as you can see, it's a geometrical progression. So the amount increases dramatically, which helps to get a signal. So this is how a signal gets generated in the standard RT-PCR test. So every time when there is a step of annealing and then extension of DNA, when a new copy is made, there is a fluorescent reporter being released, which is initially silenced by quencher. But then when the complementary DNA strand is formed, this link is broken and fluorescent reporter gets released and it generates a signal. And so as more and more copies are generated, more and more of the fluorescent reporters are released. So the signal is increased and eventually it can be detected. But of course, when you're talking about this dramatic amplification, first it takes time. We need to wait until it happens and we can actually detect a measurable fluorescent signal, but also it can be prone to false positive results. So what is the alternative? So of course, again, as a group that works on developing photonic sensors, we're curious what photonic sensor can do to help to improve the sensitivity of the detection. In the current landscape, there are a few solutions that have been used to detect either viral RNA or the whole virus by using, again, standard approaches in the photonic sensor design, either a surface plasma resonance that I already mentioned before in the context of methane sensing, or different type of interferometric photonic sensors. So the problem is still challenging because there may be just a few copies of a virus present in the sample. And those copies need to be captured, and that signal should be enough to overcome the limit of detection of a particular detector. And it may not be just enough of those copies, and you can never get a reliable signal unless you do that material amplification. So that's complicated. And also a lot of these sensors, they're typically referred to as label-free, which is true. So they don't actually use those fluorescent labels. But it does not entirely capture what is happening there, because you still need to have a sensitive target selective receptors. So those molecules still need to be attached to something very specific to them, captured by those specific receptors on the surface. And those need to be developed for each individual new virus or whatever molecule we're trying to capture. First, you need to develop all this chemistry specific for capturing this particular pathogen. And that takes time. For a new pathogen, it can take months to develop this new chemistry. So it would have been much better and faster to deploy new sensors if we could use the same chemistry for all of them and just modify something else. And then we can very quickly redeploy existing sensor techniques to address a new pathogen. So how this can be done? So again, in the standard configuration, you need to have those specific receptors. They capture specific targets. And if we're lucky enough to have enough of those target molecules, this generates a measurable signal, either the change in the wavelength or the change of the angle of excitation. So this is how some of these sensors may look like. So this is an example of an integrated silicon ring resonator biosensor. So it's a on-chip silicon ring coupled to bus waveguide. So the signal is coming from the waveguide, and it gets coupled to the ring. If some target molecules get attached to this ring, captured by those specific receptors that need to be put on the ring before you do testing, it changes the conditions for the light propagation and changes the resonance of the system. And this is something that can be detected. So again, the problem may be that the changes are not enough and you cannot overcome the limit of detection. So what we suggested to do is to rethink this whole idea by still using the same photonic sensors. A lot of existing sensors can be used with this new idea that we are trying to put forward now, but they need to be redesigned in how the detection happens. So what we suggest to do, and we already have some first experimental results for different photonic biosensor systems, is to functionalize the sensor surface first with existing chemistry and with some simple reporters like metal nanoparticles or quantum dots or fluorophores. So any existing label particle will work for it. And the choice may depend on a specific sensor configuration. So some may work better with metal particles, some may work better with quantum dots. But the idea is you use existing chemistry that's already developed and the same chemistry for any detection, where we use single or double DNA tethers to attach those nanoparticles to the surface. And then how the actual specific detection happens is that you also need to have a CRISPR complex. So this is an enzyme, and I'll talk in a second what it is, which can be activated by a viral RNA. And then once it's activated, it starts cleaving any other RNA or DNA strand it finds. And some of these complexes are known to produce tens of thousands of collateral cleavages after being activated by just one target molecule. So this is the biological amplification step that we are adding to the system. So you can imagine there is only one molecule, one RNA of a virus, for example, which if it activates this complex, then it starts cutting tens of thousands of reporters. And this signal is huge. So this signal can definitely be detected and it can be monitored in real time now because we're using quite sensitive detection, much more sensitive than just fluorescence detection used in RC-PCR. So this combines now biological amplification and optical amplification. So what are those CRISPR enzymes? It's just a family of DNA sequences that are found in genomes of prokaryotic organisms, such as bacteria. So effectively, this is a bacterial immune system, and their job is to detect viruses and to destroy them. So that's why once they are activated by a certain virus, they start cleaving it. And this is the biological machinery that is naturally developed, and we can just repurpose it for our applications. So there are already quite a few different CRISPR-based biosensor types of detectors on the market, but most of them are just based on the chemical processes, on this chemical amplification. But if they can be combined with the photonic amplification on a chip, then you have these two types of amplification combined, and the overall sensor performance is dramatically improved. And that's exactly what we do. So the first system we looked at with this mechanism is the same microring silicon integrated microring system I already showed you before. So we use the standard chemistry that is used for tethering reporters to silicon surface. And our reporters in this case were quantum dots. And then once we activated an enzyme to cleave those dots, we could see a clear shift in the resonant feature of our microring resonator. And this is very detectable, so it's certainly above the noise level, and it provides this required sensitivity of detection of a small amount of pathogens. So another platform we looked at is a very simple sensor platform. It's a porous silicon interferometer, which in this case, it's not integrated, so it does not need, the light is not coupled through a waveguide on a chip. It's just simply interrogated from the top, as shown here. So you can have a light source and a detector, and what you do, you measure the reflected signal, which because of the interference in the signal reflected from the top surface of this detector and the bottom surface creates an interference Fabry-Perot picture shown here. And then upon addition or cleavage of labels, this whole spectrum shifts. And again, the shift that we observed was really large, so it was easy to detect the signal. So this was done experimentally, proving that this concept works, and published a couple of months ago. And then we started looking, now in principle that we understand that this mechanism provides a strong amplification of the signal and can be combined with different types of biosensors. We tried to design a simple and most efficient biosensor specifically tailored to work with this new sensor approach. And the type of biosensor we looked at is a silicon grating. It doesn't have to be silicon, so it's any kind of diffractive type of sensor, usually based on a grating, which work in diffracting incident light into different diffraction orders. And if you can design it in such a way that when something changes in the sensor, in our case, it will be cleavage of those particles that we will attach before the test is run, then that changes the distribution of light diffraction in different orders, and that's something that you can easily detect. So that's a picture, for example, shown at the bottom here visually. So if we excite light normally, but then where the diffraction happens, it can switch from predominantly second grating order diffraction to zero grating order diffraction. And this can be easily monitored, so you don't need to trace the whole spectrum. You can simply have one detector monitoring the second grating order and one detector monitoring the first grating order. And then you can see how the energy is redistributed in between these orders. And you can even monitor changes real time when the concentration of particles is decreased because of continued cleavage. We can see the changes, and we can even measure then the dynamic of the process and the amount of labels cleaved in a certain amount of time. So this provides a new powerful mechanism, and we think a lot of other photonic biosensors can be redesigned to work with this new conceptual approach of sensing. And we're very excited about this. So this paper is going to be published in a week, probably, also in Optics Express. So if you're curious about more details, you can have a look. And the very last topic I'd like to mention today is, well, I talked a lot about how we can measure pollution already out there, whether it's viral or methane or any other type of pollution, like microplastic pollution, for example, shown in this bottom figure. But it's always better to prevent than to sense. And in some cases, we don't have much control, but in some cases we do. And for example, with microplastic pollution, that's entirely our fault as humans, that all that microplastic is there. So instead of trying to just capture it, we can design approaches to prevent it from even getting into the environment. And one approach we are taking is we are engineering new types of textiles that will be less damaging to the environment on every step of their production and use. So these textiles are based on using polyethylene to make fibers and fabrics. So this is in the picture, this is the actual fabric that we made. Can also be made into the form of shoes or anything else that's made of textiles. And polyethylene is actually plastic that's probably the most produced plastic in the planet. It just has not been used before on a mass scale for textile production. And through our research, we demonstrated that it's actually much better for both humans and the environment than any other synthetic or natural materials that are currently used in the textile industry. So we have a paper coming out also in a couple of weeks in Nature Sustainability on this. And so the problems we are addressing are all the problems along the life cycle of any textile. So textile production usually requires a lot of water. And this is just an example of water scarcity that can happen after overgrowing of cotton. It produces a lot of industrial pollution because of the way how materials are dyed. So when you're just dunking textiles into solutions of dyes, and then they need to be washed many times before the dye stops leaching. And then, of course, it all goes in the rivers and ultimately in the ocean and in our food supply and everywhere. Also, amount of heat produced both during production and when we do laundry, that leads to additional environmental pollution and water eutrophication when this algae grows dramatically. And at the end of life, also, most of textiles are never recycled for different reasons, because, for example, most of them are made from multiple materials, and they're very hard to separate at the end of the garment life cycle, and it's not economically viable to do it. So people just throw them into the dump, and they decay, and they further pollute with plastic and dyes. So what we do instead, we engineer monomaterial textile platforms that are all made from polyethylene with maybe some addition of inorganic particles that can be very easily separated at the end of the life cycle, mechanically and by centrifugation. But otherwise, we just engineer only one material, one polymer, and we do all the changes through changes of the morphology, size of fiber, shape of fiber, composition of the fabric. And we color it in a completely dry fashion, so no water is needed, so there is no pollution that happens in the process. We can use plastic that is currently not recycled, like those plastic bags that are floating everywhere, because it's not economically viable to recycle them back into plastic bags, but it makes sense to recycle them into high-value products like textiles. And also polyethylene can be made from sugarcane, bio-derived nowadays. So and that leads us to this monomaterial closed loop material cycle, which produces significantly less pollution at every step of production and use. And also through testing, we found that actually it prevents growth of bacteria naturally without even adding any antibacterial addends to it. And by amount, so if you measure in 24 hours how much bacteria will grow on different fabrics under identical conditions, so it's just shown schematically by the size of these circles. So you can see cotton is usually better than any other than any synthetic material. And interestingly, our synthetic polymer polyethylene performs as well as cotton, which is really nice. So it provides this natural protection against bacterial spread. So that could be quite handy again in the current situation. So this is again just showing the life cycle of this product. And I have a question if it's possible to design the sensors using the washable fabrics. So in principle, yeah, there are techniques how sensors can be integrated into fabrics to be further washed. So in this case, again, I want to reiterate, this is not a sensor. This is just a fabric technology that produces less pollution. So then you don't need to sense for the pollution because it will be less of it happening. So this is a preventive feature rather than a sensing feature. Okay, and this is pretty much I'm coming to the conclusion of my talk. So I covered briefly several topics, and if you're interested in learning more, please either email to me, ask me now, of course, or look up in our website for any additional information. So thank you very much. Again, I appreciate the opportunity to speak to you today, and I'm welcoming any questions.
Yeah. Thank you, Svetlana.
Thank you.
Yeah, we have a couple of questions. You were mentioning about SVTEX, which can reduce bacterial growth and also the spread of infection by microbes. Right. So how do they help in controlling bacterial growth? Like what is the actual mechanism or which component has the antibacterial activity?
We're actually not entirely sure. So that's something we're studying right now. We think it's connected to the very high crystallinity of polyethylene. It's typically higher crystallinity than other synthetic polymers. And it's been already known from like studying how polyethylene decays in the environment. It decays slower than anything else because bacteria doesn't like to eat it. And so it's resistant to that. And we saw that, well, that's not good when it's already in nature, but it's actually great when you're still wearing it. So why don't we use this effect? And we did testing, and it does have an effect. So we're now trying to re-engineer fibers with different crystallinity level and see if that has a noticeable effect, if it correlates with the antibacterial protection. But I don't have a definite answer why. We are not adding anything to it. So it's just a polymer by itself.
Okay. And the next question is, how are optical-based sensors better than other sensing methods?
Well, it depends. For a specific application, they may not be better. But just right now, because there are already techniques to mass fabricate a lot of these sensors, like, for example, integrated silicon photonics, there are multiple foundries around the world which can very quickly fabricate millions of these sensors on a chip, because each silicon wafer can actually host hundreds of thousands of those sensors because they're so small. So that could allow this massive scale up of the technology when it's needed to be tested daily. And it can be done on relatively cheap nowadays if you count it like per sensor. So optical amplification is remote, it's non-destructive. So if we're talking about, for example, marine sensing, something where you cannot really reach and you don't even need to collect that sample in some cases. You can just immerse something in the ocean and then measure continuously, monitor the optical signal coming through fiber. So there are multiple advantages, but in some cases, it could be that maybe optical sensor is not the best solution for a problem. So I'm not saying it should replace all the other sensors.
And one more question, how to detect dissolved methane in seawater?
So how we do that, it's so there are certain molecules that selectively trap methane and not other types of gases present in the environment. And these molecules can be incorporated within a polymer matrix, which is permeable to water. So then you immerse it in the environment, in water, and you wait a little bit, and then methane gets, so water gets recycled through this plastic, and methane molecules are captured. And then eventually they occupy all these capture sites, and that changes the refractive index of the polymer. It changes it very, very little. It's like 10 to the power minus 4 change in the refractive index. And that's why we still need a super sensitive optical scheme to detect that small change, but it is doable.
Yeah, thank you. Thank you, Svetlana, for accepting our invitation.
Thank you for inviting me. It's a pleasure. Okay, I'll stop sharing.
So good evening, everyone. The next session is going to start in a while. Please stay online. So in 2 to 3 minutes, we will start the next session.
Thank you, Dr. Svetlana, and hi, Dr. George. Yeah, this is Grace here. I hope you joined.
Yeah, I'm here.
Yeah. Hi. Yeah, this is Grace. So thank you once again, Dr. George, for accepting the invitation. I think we can get started with your presentation. Yeah. Yeah, so for this session, Professor George Jacob will be chairing your session and he'll be introducing you to the participants. And after that, you can share your presentation.
Great.
Yeah, thank you. So,
Thank you.
Thank you.
So can you see my slides?
Yes, yes, George. But just before that, we'll introduce you, and then after that, you can start sharing your presentation.
Okay.
Yeah, just give me a couple of minutes.
Yeah, yeah.
Yeah, thank you. Yes.
Good evening to all. I would like to introduce George Malrias. George Malrias is the Prince Philip Professor of Technology at the University of Cambridge. He received a BS in Physics from Aristotle University, Greece in 1991, and a PhD in Mathematics and Physical Sciences from the University of Groningen, Netherlands in 1995. After postdocs at University of Groningen and at the IBM Almaden Research Center, California, he joined the faculty in the Department of Material Science and Engineering at Cornell University, New York in 1999. From 2006 to 2009, he served as the Lester B. Knight Director of Cornell Nanoscale Science and Technology Facility. He moved to the Main Saint Etienne, France in 2009, where he started the Department of Bioelectronics and served as a department head. He joined the University of Cambridge in 2017. Professor Malrias' research on organic electronics and bioelectronics has been recognized with awards from the New York Academy of Science, the US National Science Foundation, the DuPont, and with an honorary doctorate from the University of Linköping, Sweden. He is a fellow of the Material Research Society and the Royal Society of Chemistry and serves as a deputy editor of Science Advances. He is the director of the Engineering and Physical Science Research Council, interdisciplinary research collaboration in targeted delivery for harder to treat cancers. Professor Malrias is a co-author of 400 plus publications in peer-reviewed journals that have been received around 32,000 plus citations. His current H-index is 97. With this short introduction, I would like to invite George Malrias to deliver the talk.
Thank you very much for the kind introduction. Thank you very much for the kind invitation. It's a pleasure to be with you, albeit virtually because of the times we live in, but I hope to get a chance to visit in the future and meet some of you in real life. So let me share my screen. Can you see the slides?
Yes.
Excellent. I think there should be a way to get a pointer here. Yeah, there it is. Great, wonderful. So thank you again. We'll talk about electronics on the brain. And what I want to do is to go over the field of bioelectronics and explain some of the prospects and challenges, and then show you a couple of examples of work from our laboratory, a more older piece of work, and then something more new and not yet published. So what is the motivation of this work? The motivation is to help people who suffer from neurological disorders. And neurological disorders are the leading cause of disability worldwide, with over 270 million years properly adjusted for disability, lost to stroke, migraine, dementias, epilepsies, and injuries of the brain and the peripheral nervous system. So this represents obviously a major societal problem it's a tragic thing that affects many families. What can we do about this? Unfortunately, the answer to this is not going to come in the form of a new drug. The chances of a new drug that cures Alzheimer's disease or stops drug-resistant epilepsies or repairs the brain after trauma is very low. And the reason for that is that drug discovery is becoming slower and more expensive as a function of time. So developing a new drug is a game of diminishing returns. This has been baptized as Irum's law. Irum spells more backwards. Moore's law in electronics tells us that every year the newest computer will be more performant than the model the year before, and it will cost less money per unit of processing power. So things accelerate in electronics. Unfortunately, in pharma, it's the opposite trend. Things are slower and more expensive. However, there is good news, and the good news comes from the field of bioelectronics and what is today called bioelectronic medicine. One example of this is deep brain stimulation, where electrodes are implanted in the brain in a structure shown here that's called the subthalamic nucleus. And through stimulation by current pulses, you can ameliorate the symptoms of motor neuron disorders, as shown here. Dystonia, Parkinson, tremor are conditions that are being addressed by this. This has been extended to epilepsy, where it has been shown to have beneficial effects in the frequency and severity of seizures. It is being used in neuropsychiatric disorders such as obsessive compulsive disorder and depression. It has even been tried to help with obesity, and many more applications are envisioned. As you see here, with a flick of a switch, the patient goes from not being able to hold a glass of water to being able to function almost normally. So life-changing, amazing, incredible technology, very, very promising. Where is the catch? Why don't we see this deployed at scale? And there are, despite the promise, there are some severe limitations. First and foremost is we don't understand how it works. To begin with, we do not understand how the brain works. There are over 80 billion neurons in the human brain that form networks and networks of networks. And although we understand how one neuron generates an action potential, how this action potential is propagated down its axon, arrives at the synapse, and then either excites or inhibits the postsynaptic neuron through the secretion of neurotransmitters, we do not understand how we go from this molecular type of phenomena or cellular type of phenomena to macroscopic behavior in the organism. There is a big gap there that isn't filled in. In order to understand this, electrophysiologists use electrodes that are either penetrating into the brain or placed on the surface of the brain or placed on the scalp to study the electrical activity of the brain and correlate behavior of neurons to behavior of the organism. The opposite is also not understood. What is the mechanism of neuromodulation? Why, when I apply a current at an implanted electrode, do I change behavior? What are the phenomena that take place at the cellular level as a function of time that give these changes of behavior? So these fundamental issues are associated also with technological limitations. The signals that we're trying to detect are small and diverse. They, at the first entry, you're looking at electrical activity caused by a sea of small metallic ions, sodium, potassium, that are set in motion when neurons are firing. 2 biochemicals that you want to detect at the synapse or to understand the homeostatic processes in the brain. 2 more complex cellular effects and require a diversity of sensors to be placed into the brain. The environment is hostile to electronics. When you put a probe into the brain, it's not bad only for the brain, but it's bad for the probe as well. The cerebrospinal fluid can be quite corrosive to a lot of electronic materials. The environment is highly invasive, obviously, and multiple surgeries are often required in order to replace corroded leads or exhausted batteries, or just move the probe to a slightly different location. So it is good to be a technologist in this field because new technologies are desperately needed in order to address these limitations, to overcome these limitations, and enable a better understanding of the brain and enable bioelectronic medicine. So if I were to recast the field, you can recast it as an exercise in bridging differences. For example, living systems are made out of tissues, especially the brain, that are very soft, mechanically speaking. Their communication is complex, whether this is the communication between cells, tissues, organs. The signals that are emitted are very complex and diverse. Moreover, biological systems are dynamic. This little fellow will double in size and his brain chemistry will change. That is an aspect that is not captured in electronic systems. So current electronics, traditional electronics, are made out of mechanically stiff materials with a modulus, young modulus that is several orders of magnitude larger than that of the brain. The communication between 2 components takes place by exchanging electrons or holes, something that we understand in a quantitative fashion. And electronics are static. They spend their lifetime in the form factor in which they were fabricated, save for some more recent work on bioresorbable devices. So you can discuss bioelectronics as a field, as an exercise in matching properties, in making electronics that are soft, complex, able to communicate with complex signals, and dynamic so that they better interface with the tissue. This way, though, of viewing the field is great for guiding materials research and the development of new devices. However, it lacks an explicit focus on the end result. And the end result would be to take technology and transfer it to the clinic at scale. So another model is more instructive in this, under these considerations. And this is called the push-pull model, where you can say in a typical science/technology laboratory, we would have expertise that are relevant to the clinic. For example, in my lab, we have expertise in organic electronics, in microfabrication, some other areas. And this creates a push, a science and technology push. It's like having a hammer and you're looking for nails. At the other end, at the clinic, you have needs that are well identified. For example, in epilepsy, we know that we need better arrays for mapping the brain at higher precision. And these needs create a clinical pool for new technologies. So you can view bioelectronics as a field, as an exercise in building a pipeline that will take new technologies and bring them to the clinic. And vice versa, that it would take the the needs from the clinic and inform the development of new science and technology. So you're talking about setting up a system of in vitro models, in vivo models, ex vivo models to arrive in humans, and vice versa, organizing specialty clinics or looking at priority setting partnerships for guidance for the transfer of needs to inform the technology to the clinic. And of course, this has to be done in an environment that includes all stakeholders. You have to be talking to patients, regulators, ethicists, industry, and so on and so forth. So I think this way of viewing bioelectronics is very powerful because it explicitly considers the end result and focuses on impact. A ramification of this is that you start pondering the question, what does it take for a new technology or a new material to move from the lab and make it to the clinic? And what it takes is compelling advantages. In order to bring a new material or a new device to the clinic, it needs to have advantages that are not just 10% better than what is there, but considerably better, so that you make a compelling case. The reason for that is every time you introduce novelty in the clinic, you expose yourself to legal liability. In addition to that, you have to front quite a large cost that the companies that follow you do not have to pay. So that's the reason why the compelling advantages are needed. So I will show you one example of a technology that has made it to the clinic from the lab. And this is a couple of years old. And then I'll show you a new technology, which is yet still in the lab, but I think it's on its way to take the same path. So the first example is what is today called the neurogrid, which is a very high resolution cortical array for mapping the brain. Now, this is a well-identified clinical need. This is the current clinical standard for mapping the brain. You use arrays that have quite large electrodes, many millimeters in diameter, made out of platinum, that are inserted under the dura in order to map activity on the brain. And the spatial resolution is very poor, is roughly defined by the distance between the two electrodes here. And it's centimeters because they are, you need to have large electrodes in order to record the very weak signals that emanate at the surface of the brain. The reason why the signals are weak is because what you're trying to do is to couple electronic charges in an electronic material with ionic charges in the cerebrospinal fluid. If you try to do this with silicon, you have typically an oxide or a nitride that separates your electronic material from the source of signal here. And as a result, decreases the coupling between electronics and ionics. So people use metal electrodes made out of platinum, gold, that do not have a barrier layer. And then you can have intimate coupling between the two charges, as shown here, along an interface. However, this has a limited capacitance, which measures, again, the coupling between ionics and electronics. And it's very difficult to overcome that limit. However, there are materials that allow transport of ions in the volume of their bulk. This is an example of a conducting polymer called P.PSS. It consists of two polymer chains. It is a degenerately doped P-type semiconductor that is able to uptake ions in its volume. So it's a mixed electronic ionic conductor. And in this case, you can have coupling of electronic and ionic carriers in the whole volume of the film. And that gives you a capacitance per unit area that largely exceeds the double layer limit and allows you to make novel or state-of-the-art performing devices. the manifestation of this volumetric coupling is shown here. If you measure the capacitance of a P.PSS electrode, you will find that it scales not only with its area, but also with its thickness. So it scales with the volume, which means that ions can penetrate and distribute within the volume of the film. The ramification of this is that if you have a film of just a bit over 100 nanometers thick, you can get capacitances per unit areas that are 2 orders of magnitude larger than what the double layer capacitance limit tells you. And as a result, you can now make electrodes that are much smaller that can record neural activity with higher precision. So in a sense, you gain in spatial resolution. This is shown here, where we microfabricated P.PSS electrodes to make them the same dimensions as the soma of neurons in the brain, same order of magnitude. And we integrated those with flexible, thin plastic substrates so that they can follow the curvature of the surface of the brain and make good contact. So as you see this electrode, this is a denser array that has 10 by 10 micron square electrodes. And you see that it conforms nicely to the shape of an orchid leaf, showing radii of curvature that are appropriate for sitting on the brain. And then when this was placed on the cortex of a rat, we were able to measure this spiking activity that corresponds to single neurons being picked up by different electrodes. So in a sense, you reach the ultimate spatial resolution. You reach the limit where you can pick up now individual neurons without penetrating the brain, by just sitting at its surface. This is very powerful because you go from centimeter spatial resolution down to single neurons. You can now latch into a single neuron and follow its chatter over time. It's a great tool for neuroscience. But it's more than that. It's also a great tool for the clinic because it allows you very high resolution recordings. And as a result of this compelling advantage, the fact that you go from centimeters to tens of microns in spatial resolution, this is now used in the clinic. The first introduction, the first in human trial was in 2015. And since then, many clinics have been adopting this technology currently for intraoperative recordings, and hopefully in the future for chronic recordings. And again, in humans, you would see the spiking activity that would correspond to single neurons. And at the same time, you measure more average activity that corresponds to the chatter of many networks that is called local field potential. So this, I try to address a bit of a broader view. In a lab where you develop technology, you have to also worry about, do I learn anything that I can show as a physicist or as an engineer? What are the advances to my own field? One of the things that was a consequence of this work is that we learned that there is no good understanding on the physics of electrochemical doping. In these materials, we need to get ions from an electrolyte and put them into the bulk of the structure. And we don't understand how their energy levels change. So understanding the physics of electrochemical doping is a field that will benefit tremendously from the development of these devices. Let me show you now another example of a technology that is very close to being transitioned out of the lab. And this is in the big picture here is devices that change shape, that do not have a predetermined characteristic shape, but they can adopt different shapes during their operation. So the clinical need for this particular device is in less invasive pain stimulators. For people with chronic pain, a treatment option is to put a paddle electrode, as shown here, on the spinal cord and electrically stimulate. And that has been shown to replace the sensation of pain with a sensation that is more agreeable. In order to place these paddle electrodes, a laminectomy is required, which is quite an invasive surgery that requires generalized anesthesia and requires a lengthy stay at the hospital. As an alternative, you have this percutaneous linear arrays of electrodes that can be placed in the spinal cord using a spinal tap, which is an outpatient procedure. It doesn't require generalized anesthesia. But unfortunately, these devices are not as efficient in stimulating as the paddle electrodes, and they can shift around during the life of the patient, losing their efficacy. So a patient today has a choice: a device that works well but requires brutal surgery, or a device that is easier to place but it's not as effective and might shift out of place. We were very much inspired by the field of soft robotics and by demonstrations such as this one, where you have a material that changes shape upon pneumatic actuation. So this is a system where you blow air into it and you can change its shape from flat to curvy. And as a result of this, it can walk. So we started introducing microfluidic channels in flexible electronics and experimenting with ways to achieve actuation, such as unrolling in an axial direction or in a lateral direction. And trying to figure out what can be achieved by this. And here, if you can take a jump into how you view electronics, a piece of electronic that has this embedded microfluidics could be changing shape. So it could adapt to different shapes or have a pre-programmed shape that it would deploy into upon actuation. Here's one example. You could make a paddle electrode that would normally require a laminectomy to be placed. However, in this case, because it's made out of flexible electronics, you can roll it up into the shape of a needle, the shape of a percutaneous electrode that can be placed into the body through a lumbar puncture. And then using fluidics, you can deploy it into the shape of a paddle, as shown in this in vitro model here. And the impedance will not change when you go through this. With flexible electronics today, we know how to design electronics that are robust to mechanical torture. So these are tested in cadavers. This is shown here in human cadavers, the implantation using a spinal tap. Here is the device. It has two markers on the bottom and the top end of the device, that when the device is rolled, they are brought together as shown here. And when the device is then deployed, they show the magnitude of the deployment by shown here. And the reason why there are two markers is so that you know exactly which direction the device went into the body as well. So we have shown this to work in human cadavers. We can implant and deploy these devices at the right location. And now this technology is taking its translational pathway. The next level would be in veterinary patients to be deployed in companion animals that suffer from pain after injury in order to see if it has efficacy that can be translated to quality of life of these patients. This is a device that is deployed on the brain. I showed you earlier a picture of an ECOG, a cortical array that required a quite massive craniotomy to be placed. Here we just have a small burr hole. We deploy the device and then expand it to 2 centimeters distance while it's sitting on the cortex. Again, minimizing the trauma, minimizing the invasiveness of neural surgery. So again, here, what's the science? You do that, you translate the technology. It's a wonderful thing. What would be the impact in science and engineering disciplines? There are lots of questions that need to be addressed. For example, what is the optimal device geometry and folding pattern in order to get the right deployment that you want? And it also begs the question, can you envision self-motile devices that will go into the brain or inside the body and then change shape so that they can move on their own autonomously and go and implant the right place, following non-straight paths that are not accessible surgically today. So there is a lot of work to be done. But what I hope that I did is that I convinced you that the field of bioelectronics, or bioelectronic medicine as it's called, represents a promising technology for addressing neurological disorders and beyond that, human disease in general. We talked about the push versus pull model that helps focus research carried out in academic labs on impact, and impact being translation of technologies to the clinic. We talked about the need for compelling advantages that are needed in order to translate a new technology or new materials, new devices to the clinic. I gave you an example of the neurogrid, where the compelling advantage was a spatial resolution that went from centimeters to tens of microns. And then I concluded with an emerging theme. These are devices that change shape upon pneumatic actuation. The demonstration here was a device that goes from percutaneous to paddle shape electrode and can help avoid a laminectomy in spinal cord treatment of pain. So with that, let me thank my group. Damiano Barone is a clinician with whom we co-lead the lab, and he has been instrumental in helping us focus on clinical impact. Collaborators at Cambridge, outside Cambridge, funding organizations who funded this. And again, thank you very much to the organizers for inviting me, and thank you all for your attention. Are there any questions?
Hello.
George, are we audible? Actually, Professor George is asking questions, sir.
No, I cannot hear anything.
Oh.
Am I audible to you?
Yes, now you are. Yeah.
Oh, sorry. Thank.
Now I lost you again.
Hello.
Yes, now I can hear you.
Yeah, thank you, Maldias, for this wonderful talk. It's a real example for how the nano engineer can work with the medical practitioner. Okay, so I have a few questions. if we use a physical sensor for brain mapping, how to decide this spatial resolution of the sensor array? Is there any specific techniques, or we will randomly place?
So it depends on what you want to do. For example, if the objective is to drive a prosthetic arm, you would place the array over the motor cortex in the area that corresponds to where commands would emanate for that limb, for the arm or for the leg. If you want to provide sensory feedback to the brain from pressure sensors mounted on a robotic arm, you would go on the somatosensory cortex. So the location of the implant is roughly known in the normal brain. Now, in terms of spatial resolution, again, in the context of brain-computer interfaces, there are two schools of thought. There is one school of thought that says, let's imagine that you're trying to make a highly articulate prosthetic arm. Let's try to identify the neuron or neurons that issue the command for this motion, this little motion here, and then for this motion, and then for this motion, and try to interface with those. So when these neurons fire, then we know which motor on the prosthetic arm to drive. That school of thought requires making connections with single neurons, so very high resolution implants that are stable over time. This is a tall order. It's a very difficult thing to do. And that's why we don't have such highly articulate arms. And other school of thought says, rather than looking at such low-level information, let's look at higher level information, such as volition. For example, let's look at the activity that corresponds to the thought, I want to move my arm and grab that glass of water. And then let's use machine learning for the robotic arm to learn how to make this motion by itself, rather than every little motor of the arm moving by direct control to the brain. Let's outsource that. You just give to the robotic arm the command, grab that glass of water, and then the robotic arm does that autonomously. So I think the jury is still out as to which model is best. They both have their merits, and it's very interesting to see how they will develop.
Thank you, George. Are there any other non-invasive techniques for studying the brain mapping?
Yes, so there are the field of electrophysiology, I think, goes hand in hand with the field of brain imaging using MRI. And this is typically practiced in the whole brain context. A friend of mine told me once a very nice paradigm. Imagine that you're an alien, that you're looking at a new planet and you're trying to understand how it works. How do people in that planet behave? If you're an electrophysiologist, you are listening to their telephone conversations. If you're an imaging person, you're looking at what they eat. So you're looking at metabolic activity. These schools of thought run in parallel and they don't talk too much. So I think there is tremendous amount of mileage to be had in combining electrophysiology and metabolic measurements. Now, this would require the development of better tools, either electrophysiology tools that are transparent to MRI and they do not disturb the MRI signal, the MRI imaging process, or local metabolite sensors that can be integrated with electrophysiology probes.
Thank you. There is a question from our audience. Can you suggest what type of materials are used to manufacture this type of sensors?
So for the active electrode, we like to use a conducting polymer called P.PSS. It's a commercially available material that was developed initially for anti-static coatings for applications later in displays and thin film electronics. And it's quite useful for bioelectronics. It's not the best performer. There are synthetic materials, there are materials being synthesized today that perform better, but not compellingly better. P.PSS is in the clinic. It has FDA approval in a catheter. When it's placed in a catheter, the catheter is FDA approved. So it's a major advantage for using this. One issue with the field of organic electronics is the following. It's a bit like playing with Legos. You can very easily manipulate the structure of the molecules and study structure properties relationships. This is wonderful if you're a material scientist. You can have an infinite variation in the molecular structures and understand a lot about the science of organic semiconductors. However, when it comes to commercialization of devices or translation to the clinic, this big advantage all of a sudden becomes a big disadvantage, because what you want is to focus your effort on one material and have many years of human study of that material. And if you have 1 million molecules that are possible, then the effort is spread along a quite large area.
Thank you, Marius. And the attendees asked what is the degradation rate of this material? How fast this material will degrade?
Sorry, how far it will be? What?
The degradation of this material, whether any degradation is possible or that it will affect any issues in the future, or clinical failure in the future.
So no, it doesn't degrade. It doesn't, it's foreign to the body. So the body doesn't know what to do with it. It cannot chew it up. There are people who work with making these materials biodegradable by introducing links along the polymer chain that can be cleaved by enzymes to make the material degradable. But by and large, these materials are not meant to biodegrade, which is desirable in a lot of chronic implant applications.
What is your opinion? Is it possible to use magnetic materials as a bioelectronics material? Is it possible to use magnetic material as a bioelectronics material?
So there are magnetic materials used and magnetoelectric materials used as both sensors and applications. Yes, there are some recent papers on magnetoelectric materials for use in implant applications.
Okay. Thank you.
Yeah. So if there are no other questions, thank you once again, Dr. George Milleras.
Thank you.
Yeah, for accepting this invitation. Thank you very much. Have a nice day. Thank you.
Thank you all. Bye
Yeah.
bye.
Bye bye bye.
Thank all the participants and speakers for today's talk. I request all the participants to join for tomorrow's talk with the same link. So we'll meet tomorrow at 8 a.m. in the morning. So thank you. Have a nice day.